Welcome to the first event in our 2024 Science Series. For those of you who came to our events last year, you'll know that this is intended to give an overview and introduction to life sciences technologies, hopefully to assist those of you advising your business in relation to those technologies. Today we are going to have a session on antibody-drug conjugates and radiopharmaceuticals. Really exciting because they're both super interesting technologies that are really taking off at the moment. And our speakers today will be Claire Phipps-Jones in the patent litigation team, Louisa Jacobs in the commercial IP team, and Jamie Hatzel in our regulatory team. So I'll hand over to them. Thank you for coming.
Excellent. So our agenda, as Charlie has alluded to, we're going to start with antibody-drug conjugates, which I'm going to take you through. We're going to talk about what they are, how they work, some of the components to those drugs, and overcoming some of the challenges. Louisa's then going to talk a little bit about radiopharmaceuticals. And again, as Charlie mentioned, we're talking here really at the basics. So we're starting with the basics of radiation, radiopharmaceutical carriers, and then diagnostic applications. And then Jamie's going to pick up the end with some more commercial and legal considerations. So starting with antibody-drug conjugates. I thought I'd start off with what they are. Really, they're one of the fastest growing cancer medications. They were developed primarily to address the issues that exist with existing cancer therapies like conventional chemotherapy, which have off-target effects. And so here I've put on the slide, they're a guided missile. And that's really the idea, to have a much more targeted therapy. They deliver a therapeutic agent directly to the cancer cells, and they comprise three key elements. So the first part is an antibody. That's mainly to allow the ADC, I'm going to refer to them as ADCs just because otherwise I'm going to stumble over antibody-drug conjugate continually. So that allows the ADC really to know where it's going and to target the appropriate cell. They have this linker section, which is actually quite important for the stability. It needs to make sure that the drug, or the payload as I'm going to refer to it, gets to the right place and isn't dropped too soon. And then you have, lastly, the payload section, which in a number of, at least the earlier versions of ADCs, were anti-cancer drug or cytotoxic agent, but we can talk a little bit more about what those payloads may be. Now, while those are the three main elements of an ADC, I think if you're involved in developing these drugs, you need to also think about the antigen, what it is that ADC is binding to, what you're targeting to, and also the conjugation method, so how you link the linker and the payload to the antibody. So I'm going to talk a little bit about those elements. But first, how do they work? As I say, one of the things you've got to think about is this part here. This is intended to be a cancer cell, and here you've got what is referred to there as a tumour-associated antigen. It's a protein that often expresses, for example, on the outside of a cancer cell. That is the antigen to which your antibody targets. It's important to make sure that you're targeting the right thing to get the therapy into the right place in the body. And the properties of those is quite important. So obviously, you have to be able to identify them in the first instance. You don't want something that is expressed more widely in the body or in different cells in different locations. You don't want something that's going to be present in the blood because you don't want binding to happen outside of the target location. And also, it's quite important that those things don't have to be proteins expressed on the outside of a cancer cell. They can, in fact, be anything within the tumour microenvironment. So I'm going to refer to them by reference to a cancer cell and an antigen on the surface of a cancer cell, but the concept applies more broadly than that. So zooming out a little, this is the same schematic that we saw on the last slide. So we've zoomed out a bit, and this is supposed to be the outside of a cell. So what you can see is that once bound to the antigen on the surface of the cancer cell, the ADC itself is internalised into a lysosome. And this is really where the magic happens. So there's a different microenvironment inside those lysosomes, which result in the ADC either dropping or the linker breaking the payload, or alternatively the antibody falling apart and allowing the release of the payload. So therefore you have the payload, the chemotherapeutic drug, inside the cell. There's also some really interesting opportunities to take advantage of what's known as the bystander effect. So once that drug has been released, if it has the right properties, or alternatively once a cell dies, a certain amount of that drug will release from the cell that has originally been targeted and have an effect on neighbouring cells as well. And it's important to bear in mind that if the drug is still available to do that, it's not necessarily going to have an effect on neighbouring cancer cells. It could also have effects on healthy cells. So it's, in some ways it's a bonus because it can target neighbouring cancer cells, but in other instances it's a side effect or an issue that can cause a side effect. So looking at each component part up close, and I am assuming a certain amount of knowledge of antibodies, so apologies if I skip over this quite quickly. But this, on the slide here, you have an IgG, or an immunoglobulin G. And on the left-hand side is the schematic that we're going to use for the purposes of the presentation, but on the right-hand side, that's really the crystal structure, so it gives you more of an idea of what these things look like. And for binding specificity, the most interesting part of the antibody is up here, like our two little hands, which are these pockets up here that you can see. And those are the areas of the antibody that have a huge amount of amino acid variation, which allows them to be developed to have to be very specific, to have a high degree of specificity to what they bind. So that portion of the antibody is really important for specificity. We also are interested in the Fc region. That's the sort of rest of the structure that sits down here. That in the normal sort of cell environment has an effector function. Now, when I say an effector function, I mean that portion of the antibody has the ability to stimulate your body's own immune response. And depending on the antibody that you choose, the effector function that antibody has may be different. And there are also ways in which that part of the antibody can be altered so that it has less of an immune or effector function if you wanted that as well. So I've used the example of an IgG, and you'll see that throughout the slides. That's primarily because it's the most common type, but they do have their disadvantages. They're really quite big, but they are also highly specific and have a lot of useful effector functions. So looking at the desirable features of the antibody, well, first, we've spoken a little bit already about specificity, and I think that's obvious. You want to make sure that you're targeting exactly the right thing. Another key quality is binding affinity. Now, you might think you want your antibody to bind as tightly as it can because it gets internalized then into the cell, and that's all really great. And that may be true, but there are certain circumstances where actually a little bit less binding affinity may be good. So think about, for example, a tumour cell where the cells are packed quite tightly together. If you have something that binds incredibly strongly, it tends to target the outside of that tumour cell and not really permeate inside. So what you might want is to balance or to think about the level of binding affinity the antibody has so that it targets the right cells in the right places at the right time. A couple of other properties. You want to have an antibody that facilitates efficient internalization. As I say, those antibodies get captured and brought inside the lysosome. So whatever your properties of antibody that your particular antibody has, it has to be able to do that. You want to have something with low immunogenicity, and I think this is pretty standard from therapeutic antibodies. You want a human or humanized antibody, ideally. Early versions of ADCs, I think there's at least one. This is a chimeric antibody, but really the focus is on human or humanized antibodies. And you want them to have a long half-life. And as I said, depending on the choice of antibody, if you have an IgG1, it might have a longer half-life than, say, an IgG3, but there are choices available to you. But obviously that's going to affect how frequently you need to dose and things like that. So it's also worth being aware that it isn't necessary to use the whole antibody. Now, the earliest ADCs, most of them did use IgG1. And those have a lot of benefits, including the longer half-life, but they also have disadvantages. For example, they're a bit too big. They can't necessarily get into certain compartments. They can't necessarily get within the tumours. And so people started to think about, okay, how do we use the specificity of the the specificity of the antibody, but actually allow it to have more penetration. So you start to think about fragments. There are just some of the versions of the fragments that you can use that are shown on the screen. But that's really, the idea is to, as I say, increase tumour penetration and penetration into the right compartments in the body. The downside, of course, is that they may have a shorter half-life, but these things can, it's all part of a balancing act in choosing your antibody structure. So thinking a little bit about the linker. As I said, the linker itself, it's a really important part of the ADC. because that is the part that holds the antibody, which does the targeting, to the drug, which has a cytotoxic effect. And what you don't want to do is drop your payload too soon. So there are a number of different types of linkers that you can use, and primarily there are two sort of categories which you can divide them into. You can have a cleavable linker, and those are linkers that take advantage of the environment within the lysosome. So once that ADC gets internalized and it's in that bubble that you saw on the slide, there's a different environment. It's quite acidic, and there are different enzymes that are available, and they may be used to break your linker, allow the payload to roam free. Another alternative is to have a non-cleavable linker. And in those examples, what's happening instead is that the whole antibody is breaking down, and at that point in time, the payload is released. So then looking at the third component, it's, as I say, we refer to it as the payload here, or the cytotoxic drug. Most of the early ADCs carried these cytotoxic agents, chemotherapeutics and the like, and they either targeted microtubules, which contain a lot of tubulin, and if you have a tubulin inhibitor, by way of example, that is going to have a much greater effect in a rapidly dividing cell, like in a fast-growing cancer. And so that's why microtubule targeting ADCs formed some of the early drugs, alongside DNA targeting ones that essentially cause cell death by damage to the DNA. There are others available as well. I mean, for example, there are some research ADCs that I've seen using RNA targeting, but that's, as a category, those I think are the two main versions. The whole other category is this immune stimulating ADCs, or what are referred to as ISACs. Now, those act in a different way. So rather than your antibody binding to the antigen, pulling the payload in proximity and inside the cancer cell and then killing the cell, what these are doing are bringing the antibody and the payload in proximity and then recruiting and activating your own immune system. And then it's your own immune system that will then take the next steps in attacking the cancer cells. It's quite an interesting way to turn cold tumours into hot tumours. That's the way I've seen it being described, and there's quite a lot of research in that area. I'm not going to talk about radioactive payloads. That's Louisa's gig, but they can also be radioactive. Spoiler alert. And there are other, like I say, a number of other sort of novel potential ADC payloads which we don't really have time to go into today. So, desirable features. One of the really great things about ADCs is because you're very specifically targeting cancer cells, you can have a drug that has a very high cytotoxicity, so picomolar or nanomolar range. You have less off-target effects, so you can, you're able to increase that potency just a little bit. Similarly to the discussion that we had about antibodies, you want something that's got low immunogenicity. You don't want something that your body's going to respond to before it even gets to its target cell. They need to be quite stable. So we were discussing the stability of the linker and the importance of that in getting the payload to where you want. But obviously your payload itself also has to be stable. It has to be stable in the normal environment within the body, but also it has to be stable within that lysosomal environment, which has the various properties that we discussed just before. You want to have modifiable functional groups. You need, and by that I mean you need to have a part of your drug that you can modify in order to link it to the antibody. Appropriate water solubility, kind of for obvious reasons. It needs to be something that can be intracellular targeted. So by that I mean when it comes along and binds the outside of the cell, it actually will get internalized. There's nothing that stops that internalization process. And also the payload itself, the drug, it has to be matched to the cancer target itself. And that sounds kind of obvious, but I hadn't really thought about it until you start looking at the various types of antibodies that are already available. So there's a whole host of therapeutic antibodies that are available that are being used to link a payload. But obviously you've got to make sure that the payload matches the cancer cell that you're treating. So drug-antibody ratio, or DAR, is another important thing. So what you will have seen in my slides so far is this pretty picture of an antibody with one drug attached to it, slightly divorced from reality. What you have in practice is a number of payload molecules, if you like, attached to each antibody. And you can control how many, the drug-antibody ratio, by considering the conjugation method. So there are two key examples of sort of more generalised conjugation methods. There's a mechanism by which you can bind the payload to lysine residues in the backbone of the antibody. So these are the ones you can see like down here. Or alternatively, you can bind them to the disulfide bridges. Those reactions are slightly less specific. You have a little bit less control over how many various drug molecules you have attached to each individual antibody, and it gives you a population of antibodies, if you like, with an average DAR. There are also mechanisms by which certain companies have been amending the sequences within the framework of the antibody such that they can very specifically conjugate the payload to specific parts of the antibody molecule. Seems to be quite an interesting way in which you can control exactly the DAR and also ensure that your drug is binding to the antibody in places where it's not affecting either the effector function or alternatively the binding. So why do we care about DAR? One reason is there are a number of cancers where you have lower levels of those antigen expressions on the cancer cells, right? So it may be that you can have an antibody that targets a particular antigen, but even then you're not getting enough of the agent into the cell. Whereas here, if you can put multiple drugs onto each antibody, you have, even if you've got a low expression, you can still then get sufficient drug into the cell to have a more profound effect. You can also increase your bystander effect. And a tumour is often a good example of this because if you are able to get more drug into the cell and a greater bystander effect, you can have the single target or the single ADC targeting one cell, but then also having an effect on the surrounding cells as well. But of course, as we discussed before, the bystander effect, there is a potential for increased side effects. So there's a balance in all of this. So some challenges. Now, I am by no means going to cover them all, so there are just a couple of highlights here. The first is tumour heterogeneity. They're all different. They're different between patients, they're different between types of cancer, and even sometimes you have one type of cancer and different cells within the same tumour. And so for that reason, it becomes very difficult to target because you think you might have the therapy that targets one of those cells very, very well, but it's not necessarily going to be the answer. It can result in drug resistance. So, for example, one clone of cells can survive the treatment and then multiply, and there's no full treatment there. So, drug resistant, as I say, the tumours can grow resistant to ADC therapy through various mechanisms, and these are some examples of the mechanisms by which it's thought to happen. I'm sure it's not complete, but in essence, it's changes to the targeted antigen, might be mutation, might be downregulation. It's changes to the way in which the drug protein is removed. That might be upregulation of other proteins that remove the drug. It can be changes in the way in which the ADC actually gets into the cell. As I say, there are a number of mechanisms, so these are all challenges that have to be considered when developing. Yeah, we spoke a little bit about this, so low specificity, and by that we mean off-target or on-target off-tumour effects. So your ADCs can be perfectly targeted to the antigen that's expressed on your tumour. It can work perfectly. It gets internalised. It does exactly what it does to the cancer cell. But unfortunately, it also will affect a particular category of healthy cells at the same time. And so that is something that we need to be aware of. So some of the formats that are used to try to combat at least some of these issues. So we can, for example, use bispecific antibodies. And by that, I mean you have binding to one antigen on one arm and binding to another antigen on the other arm. So you're effectively targeting two antigens at the same time. That's really useful because within your tumour microenvironment or within a single cell, you might have multiple antigens that you want to bind. Or it might be on different cells within the same tumour. So that helps with the tumour heterogeneity issue. It may also allow the product to have a synergistic effect. And then last that I'm going to talk about at least, are these dual drug ADCs. So as I said, my first schematic showed you that there was just one drug. And then I said, well, actually, no, wait, you can stick loads of drugs on, all the same drug. Here we're talking about putting more than one type of drug on each antibody. So it can be really useful, particularly if you're talking about drug resistance, to have multiple cytotoxic agents on that one antibody. And then I think, as I said, there are very, very many developments in this field, and this has been very much a whistle-stop tour within my 20 minutes. But I think, for me, it's like targeting those three components, looking at the conjugation method, identifying the right antigen. These are all really key. There's also some really interesting combination therapies that are around. So, for example, using an ADC where your payload might increase the susceptibility of the cell to radiotherapy. That's quite an interesting concept. Or alternatively, using it where you're also combining it with some kind of hormone therapy. These are all options that are available and areas of interesting research that are ongoing. So it really is just a little whistle-stop tour and a glance into the world of ADCs. And then I'm going to hand over to Louisa to go on to radiopharmaceuticals.
So yes, now we're going to talk a bit about radiopharmaceuticals in this next half of the talk. So what is a radiopharmaceutical? Well, in simple terms, it's a way that you can treat disease or diagnose disease using radioactivity. And what does a radiopharmaceutical look like? Well, here's a schematic, and you can see there are a lot of similarities with an ADC, and there's some overlap there, which Claire already mentioned. So at a high level, you can talk about a radiopharmaceutical in three different parts. You can talk about the targeting molecule, which is the purple triangle there. That can be an antibody, doesn't have to be. That gets the whole thing where it's meant to be in the body, in this case, a cancer cell. And that targeting molecule is linked using a linker to a radioactive substance, and that's the payload. That's what kills the target cell. And we're going to talk about those three elements of that radiopharmaceutical in this part of the talk. But just before that, so a bit of history. Obviously, using radiation in medicine is not new. Radiation's been used for over hundreds of years in medicine. And putting radioactive substances in the body is not new. Radioactive iodine, for example, has been used since around the 1940s as a treatment for throat cancer. But with radiation therapies, off-target effects is a problem, and there can be some really strong side effects. And so one of the main goals of modern radiopharmaceuticals is to increase the specificity and minimize those off-target effects. So we're going to start with the radioactive part there and go back to a bit of basics of what is radiation. Simply put, radiation is the transfer of energy from one place to another. And we can think about two different categories of radiation. So you've got non-ionizing radiation and ionizing radiation. And the main difference between these two is the amount of energy. So non-ionizing radiation has a lower energy and ionizing radiation has a higher energy. And it's ionizing radiation that we're interested in here for this talk, because it's ionizing radiation that is used in radiopharmaceuticals. So what does ionizing radiation mean? Well, here's a schematic of an atom. So that's the nucleus of the atom, and the blue and the black blobs are meant to be protons and neutrons in the nucleus, and the red blob is an electron in the electron cloud around the outside of the atom, and the wavy line is the ionizing radiation. So ionizing radiation has enough energy to knock an electron off an atom. And when it does that, it creates a positively charged particle called an ion. And that's why it's called ionizing radiation, because it creates ions. And that can do a lot of damage in the body. And that's the power that is harnessed by radiopharmaceuticals. So where does radiation come from? Well, non-ionizing radiation is all around us. It's invisible light, the infrared on the remote control, radio waves, and that has enough energy to make atoms vibrate, but not enough to knock an electron off. Whereas ionizing radiation comes from various different sources. X-rays is ionizing radiation. And another source of ionizing radiation that's important for this talk is the radioactive decay of radioactive substances, and that's the source of radiation in radiopharmaceuticals. So what is radioactive decay? Well, you can talk about atoms being stable or some atoms being unstable, and it's unstable atoms that create radiation. So if you've got too many protons or neutrons in the nucleus of an atom, the forces in the nucleus are not balanced, and the atom will undergo radioactive decay. And it's that radioactive decay which produces ionizing radiation. And that decay happens until those nuclear forces are balanced. So a bit of terminology that we're going to use today. So a radionuclide is a radioactive atom. And that's, like I said, that's one that's got an unstable nucleus and that emits radiation. So you often, when you talk about radiopharmaceuticals, people will talk about a radionuclide or a radioisotope. So these two terms are used interchangeably often, but there is a slight difference. A radioisotope is a type of radionuclide, and an isotope, because an isotope is a form of a chemical element, and a radioisotope is a radioactive form of a chemical element. But you'll hear those two being used interchangeably. That's the payload of the radiopharmaceutical. And finally, half-life. That is, if you've got a radioactive substance, the half-life is the amount of time it takes for half of the atoms in that to decay and release radiation. And that, depending on the substance, that half-life can range from a millionth of a second to billions of years. When we're talking about radiopharmaceuticals, they use substances which decay, have a half-life of a few days, and we're going to talk more about that later in Jamie's part of the talk. Okay, so we've talked about what ionizing radiation actually is and where it comes from. So there are three main types of ionizing radiation that you'll hear about when you read about radiopharmaceuticals, and that's alpha particles, beta particles, and gamma rays. Alpha particles, so these are positively charged, they're relatively slow and heavy compared to other types of ionizing radiation. This is an example. This is actinium-225, a radioactive substance. When it decays, you get an alpha particle. And an alpha particle is actually made up of 2 protons and 2 neutrons. And that can't penetrate very far in the environment. So that could be stopped by a few centimetres of air or a sheet of paper in the outside environment. If it gets into your body, it's extremely harmful. Beta particles. These are fast, small, negatively charged particles. They can penetrate further. And they can be stopped by, for example, a sheet of aluminium. And finally, gamma rays. These are weightless. It's like a pure form of energy, like an X-ray, a bit like an X-ray. And they can penetrate really far, and as with an X-ray, you need a lot of protection in the environment against these. So to stop a gamma ray, you can have a few inches of lead or a block of concrete, and that might be able to stop a gamma ray. So these are the 3 main types used in radiopharmaceuticals of ionizing radiation. And so how does that ionizing radiation actually damage a cancer cell? Well, ionizing radiation can do a lot of DNA damage. This is a schematic of the double helix of DNA. It can cause ionizing radiation can cause a single-strand break, or it can cause a double-stranded break. But also it can damage DNA indirectly. So when we have ionizing radiation, it can react with the water molecules in our cells, and that can create highly energized particles called free radicals, which will themselves damage DNA and other components of the cell. And then all this damage can lead to cell death of the cancer cell. So that's a quick tour of the radioactive part of the radiopharmaceutical. So just to sum up, it's the part that emits ionizing radiation that damages or destroys the cancer cells. In the pharmaceutical itself, it will be protected by something called a chelator, which ensures that it's stable in vivo. We're going to talk about the carrier systems in a minute. But before I move on to that, just a note on the linker. As Claire mentioned, linkers aren't just simple linkers that links the payload to the carrier. They do more than that, or they can do more than that. So a linker may act as a pharmacokinetic modifier, which means it can affect the behavior of the whole radiopharmaceutical in the body. So it might increase the circulation time, for example. It might make the radiopharmaceutical more stable or reduce the non-specific binding of the radiopharmaceutical. So we're going to focus now on the carrier system. And we're going to talk about, very briefly, 5 different types of potential carrier systems that either are used or are being investigated. The first one is antibodies. I'm not going to spend any time on this really because everything that Claire said applies to this. Antibodies were the first carrier molecule to be used with radiopharmaceuticals, and as Claire mentioned, they've got very high affinity and specificity for tumour antigens, and they are large, and so there are issues with diffusivity in tumours, and fragments can resolve this but have their own issues. But I won't talk any more about those because we've talked a lot about antibodies. I'll talk about peptides next. So peptides as a carrier system are useful. What is a peptide? It's a string of amino acids. Amino acids are the building blocks of proteins in our bodies. And some of the advantages of using a peptide as a carrier system is that it can be easily produced, they can show minimal toxicity and have a good pharmacokinetic profile. And I'm going to use a Novartis product as an example of one of these, a product on the market at the moment, Lutathera. So this is a product used to treat a type of neuroendocrine tumour. And it consists of a peptide called dotatate, which is a string of 8 amino acids protected by and there's a chelator there as well. And this is attached to lutetium-177, and that is a beta-emitting radionuclide. So that's the small negatively charged fast particles. That binds to a receptor on the surface of the tumour cell, and that's how that peptide brings the lutetium-177 into contact with the right tumour cells. Another potential carrier is a small molecule. Now there's a lot of interest in these. These small molecules can, due to their size, they can quickly penetrate tumours, and also they can be quickly cleared from non-target tissues. And there's lots of ongoing activity related to small molecule carriers at the moment. And I'm going to use another Novartis example of a small molecule carrier drug that's on the market at the moment, and that's Pluvicto, which is used to treat prostate cancer. And it targets an antigen on the membrane, cancer cell membrane called PSMA, which is a prostate-specific membrane antigen. This is just a blown-up version of that graphic there. This is the molecular structure of that small molecule drug, and it consists, and you can see there, that's the lutetium-177, the beta-emitting particle that's in that one, the same radionuclide that was in the previous example. Nucleic acids, they're being investigated as potential carriers. So nucleic acid, DNA is a nucleic acid, RNA is a nucleic acid, they're naturally occurring. But we can also make them. When I say we, not me. One can also make them. And peptide nucleic acid, PNA, is a widely used synthetic nucleic acid, and it's used a lot in molecular diagnostics. We've shown an example here of a preclinical study that was done a couple of years ago using a PNA carrier for a radionuclide. So here you've got HER2 receptor on the surface of ovarian cancer cells. This is all in vitro. And you've got a probe here, and this green bit is called an anti-HER2 Affibody, which is a small engineered protein, and it binds this receptor, and it's conjugated to this PNA nucleic acid. And then there's a second probe, which consisted of another PNA, but this conjugated to the radionuclide, and it's complementary to that PNA. So it binds, and that's how it brings the radionuclide into the vicinity of the cancer cell. And the last one we'll talk about today, the last carrier type, are nanoparticles. So nanoparticles, this is a field that's developing as well. Nanoparticles they're bigger than small molecules. They're still very small, about 1 to 100 nanometers. They can be organic, something like a liposome, or inorganic, like we've shown here, a gold nanoparticle. And there's two ways that these can potentially target tumours. So active targeting and passive targeting. So active targeting is where the nanoparticle is conjugated to another carrier molecule. So that might be a small molecule or a peptide, anything. And that will then be specific to a receptor on the surface of the target cell. But passive targeting is a phenomenon where nanoparticles have a natural tendency to accumulate and get trapped in tumours, and that's thought to be due to the leaky blood vessels that tumours have, allowing the nanoparticles to accumulate there. So there can be a combination of the way that these nanoparticles could potentially target a tumour. So far we've talked about therapeutic radiopharmaceuticals. Radiopharmaceuticals are very important in diagnostics too, and widely used. So I'm going to do a couple of slides on the diagnostic uses of radiopharmaceuticals. First of all, what's called a PET scan, or positron emission tomography. And here we've got an example of a radiopharmaceutical, commonly used one, FDG, or fluorodeoxyglucose, which is a glucose analogue. Right, and that is attached to fluorine 18, which is the radionuclide here. And that is injected, and it's taken up. So cancer cells have a higher demand for glucose than healthy cells. So it's selectively taken up by cancer cells because it's a glucose analogue. And once in there, the fluorine 18 will undergo radioactive decay, and it produces, this is this grey dot, did you see that? A grey dot, this is a positron, which is a positively charged particle. And the positron then can collide with an electron in the tissue, and that once that collision happens, this is called something called annihilation, quite scarily. Gamma rays are produced, and those are detected by the PET scanner. So you get an image where it shows up where the cancer is, and that's the radioactivity being detected. SPECT scans, single photon emission computed tomography. Here we've got an example where the radiopharmaceutical is a sulfur colloid, which is a sulfur particles in a solution. And in this example, the radionuclide is technetium-99. And in this example, the radiopharmaceutical is injected near to the site of the tumour, and it accumulates in the sentinel lymph nodes, and these are the lymph nodes that cancer cells will first often spread to from a primary tumour. The radiopharmaceutical accumulates in those, and the gamma rays that are produced are detected by a SPECT scanner, and then this is the image that you get. And that shows the clinician where the lymph nodes are, allowing them to be biopsied and to test to see if the cancer spread to them. So wow, I felt that if that was 20 minutes, I felt that went in about 5 minutes. So that's also a whistle-stop tour of radiopharmaceuticals. And I'm going to hand over to Jamie to talk about some of the legal and commercial considerations.
So let's start with antibody-drug conjugates. If we think back to Claire's presentation for a moment, these are big, complex composite products for the most part. You have two primary active molecules involved there. You have the carrier molecule, which is doing the targeting, and you have the active cytotoxic payload as well. And we mustn't discount the importance of the linker to the overall performance of this product either. Now, because you've got so many parts at play there, it would be relatively uncommon for one company to develop every single part of this product from the ground up, which means that to some extent you're going to be relying on assets developed by another company involved. And so you've got a couple of potential development models involved there. You'll either be in licensing in a molecule, which is pretty common, and that's the kind of model that we've seen fairly frequently. Or in some instances, you might see a joint venture involved, where two companies bring together know-how and IP that they've developed to create a single ADC that they will then co-commercialize. If you wanted to think of a very, very trite example, you could imagine that one company might develop the targeting carrier molecule, and the other company might develop the payload, and then they will work together to figure out a way to best conjugate those two products to make them into a functional ADC. And there are some commercial contracting implications of both of those models. If you're going down the in-licensing route, obviously you don't want to overpay for the molecule that you're licensing in. Of course, let's imagine that you are in-licensing the targeting carrier molecule. That's very important to the performance of the product. It's not the be-all and end-all. You don't want to overpay for it. And so you want to ensure that the royalty, which is associated with the in-licensing of that molecule, is proportionate to the contribution that that molecule makes to the successful product that you produce at the end. Which means that that royalty provision that you have needs to be kept tight. And you're going to want to consider royalty stacking provisions as well. Let's say, for instance, that you are in-licensing a monoclonal antibody to use as a carrier molecule. You may have multiple layers of IP which have gone into the development of that antibody. There's going to be layer upon layer of royalties associated with that. You want to make sure that you're not overpaying. If we think about the joint venture scenario, well, great, both parties want the product to succeed. What happens when we get into our clinical trials, let's say our phase 2, and we discover that actually one part of the product seems to work very well? Maybe you're getting your product to the target, but actually then you're not having the kind of effectiveness that you want. The parties need to think about what they're going to do at that stage. Do you continue to collaborate, trying to figure out a way to fix the product, maybe license in further assets to further develop that ADC? Or in fact, do you walk away? And if you walk away, then how do you unscramble all of the joint know-how and IP that you've generated in the course of the joint venture to ensure that those assets don't get frozen in perpetuity? So moving on from ADCs, because I did promise that this was going to be brief, let's have a look at radiopharmaceuticals. Now, a lot of the same licensing considerations that I've just discussed apply here. Again, these are big, complex product, and you're unlikely to develop them entirely from the ground up. But there's a more interesting point here, I think, which is related to the science of these things. Generally speaking, radiopharmaceuticals and the radioisotopes which are used in them have extremely short half-lives. That's the rate at which they decay and cease to be effective. Taking some examples, actinium-225 is a very, very commonly used therapeutic radionuclide. That has a half-life of just 10 days. So in 10 days, essentially half of your radiation is gone. The effectiveness of that product has, roughly speaking, halved. If we look at fluorine-18, which is the substance that Louisa referred to in relation to PET scans, that has a half-life of just a couple of hours. You can imagine the manufacturing challenges which are associated with that, because your active ingredient literally starts to decay and therefore become less effective the moment that it comes into existence. And putting it in the freezer isn't going to help with that. So you need to get that product into patients as quickly as you can after manufacturing is completed. Luckily, the regulatory framework which applies in the UK and in the EU is somewhat alive to that issue in two main ways that I want to talk about. First of all, you've got some specific good manufacturing practice rules for radiopharmaceuticals, which mean that you can actually get your product into the distribution chain and on its way to the patient before you've actually completed batch testing. You need to complete your batch testing before it actually goes into a person, but you can get things on the move so that they make it to the patient as quickly as possible. And secondly, you have some very interesting, I think, specific marketing authorization rules for products which are actually used to manufacture radiopharmaceuticals. If you obtain, well, you can obtain a marketing authorization for, let me see, what's the list here? You can obtain a marketing authorization for a radionuclide generator. You can obtain an MA for a kit which is used to produce a radiopharmaceutical, and you can obtain an MA for a radiopharmaceutical precursor. Essentially all of the stuff that goes into actually making a radiopharmaceutical, but not the finished product itself. And if you obtain an MA for that, then your end user who uses that product is actually relieved from the obligation to obtain a marketing authorization for the finished radiopharmaceutical that they make using your product, provided that they are appropriately licensed in accordance with national law, and they do everything in accordance with your instructions. There are some... It's all well and good if they have that necessary license and they can use that product that you've obtained a marketing authorization for. But there's a further point which could be easy to forget, which is that holding that license doesn't relieve you of the obligation to hold a manufacturing authorization. So if in fact your end user, the hospital, is making use of your radionuclide kit, nonetheless, if they're performing manufacturing steps in the hospital, they need to hold an appropriate manufacturing authorization for all of the stuff that they want to do. Generally speaking, hospitals aren't very big fans of obtaining those kinds of authorizations. They just want to be able to use a product off the shelf. They don't want to have to engage in further regulatory compliance, further quality systems. And so sometimes you have to find an innovative solution. One thing that we've seen more in the cell and gene therapy space, but which you could imagine applying here, is what we like to call GMP in a van, where the actual marketing authorization holder for the product puts a manufacturing facility in the back of a van, parks it up in the customer's car park, performs all of the manufacturing steps on site under their own manufacturing authorization, and then very, very quickly runs it inside to get to the point of care so it can be administered to a patient. So there are some regulatory challenges here. There are also some innovative workarounds. So those are the legal issues. There's one very, very brief commercial consideration that I want to point out here, which is that it's just hard to get your hands on the radioactive material which is needed to produce these products. If you want to make radiopharmaceuticals, generally speaking, you need to get your hands on nuclear materials, and the main place in which those things are found is nuclear reactors. One of the main global sources for the products that go into radiopharmaceuticals are nuclear reactors from the former Soviet Union, to the extent that medical materials of this sort are actually exempt from the litany of sanctions which are currently applied to the Russian economy. Nonetheless, there still isn't enough to go around. Pharmaceutical companies are constantly on the hunt for new sources of these materials, to the extent that there was a recent attempted deal, which the FT reported on, to buy expended nuclear fuel from the UK's nuclear submarine fleet. That fell through, but it shows the extent to which people are working to try and get their hands on these products. Just to summarize and wrap up, there are, as we've seen, some peculiarities and oddities and challenges associated with these innovative classes of product, but none of these are insurmountable. There are ways to overcome them. And I think there's a lot to be excited about over the next few years, in particular in some classes of cancer which haven't really seen any movement in the standard of care since about the 1960s. So I'll open us up to questions.